Hello. All right. So for this video, what I want to talk about is PCR, or polymerase chain reaction. More specifically, how do you design primers for PCR reaction, or PCR experiment? We got a worksheet again to go through for this one. Just to kind of background, PCR, polymerase chain reaction, probably singly one of the most important discoveries that we've had in molecular genetics in the last half century or so. Now, what it does is we're going to do DNA replication in a test tube. And we're going to replicate a specific target area of DNA. And so what this is useful for is the genome is 3 billion nucleotides in size. We have just over 3 billion. We have 22,000, 23,000 protein coding genes, and it's a whole lot of letters. And so what we want to do is we want to be able to isolate out, say, a target sequence that we're interested in studying. Okay, and the other advantage that this allows us to do is in not only isolate out that piece of DNA or that particular sequence of DNA, it allows us to make a whole lot of it. And why that's good is what we can do is we'll then create material to be able to do downstream experiments and to be able to do other stuff and be able to determine the sequence of the PCR product, or determine the sequence of the DNA, or to use it in other applications. Lots of different ways that we can use it. It allows us to make a lot of it. Okay, because when we're doing experiments, these are small molecules, and so to do anything effective, we need really, really fine instrumentation to be able to detect tiny, tiny amounts, or we need to make a lot of what we're using so that the instruments we have that aren't really, really sensitive are able to pick them up. And so we can use PCR to make a lot of starting material. Okay. It's used in all different types of contexts, whether you see it on CSI all the time, where they're picking up the soda can and swabbing off a little bit and PCRing it and determining if it's the criminal or not. In a roundabout way, that's kind of correct. There are aspects of how they depict it that are completely wrong, but the general premise is right. You only need about 10 nanograms of DNA to actually get a product of DNA. So you can start with really, really, really small amounts and make gobs and gobs of it, millions to billions of copies of your target sequence by the time you're done. So for this particular worksheet, we're given this sequence of DNA where we have these bold texts that we're supposed to design primers for to be able to amplify those particular regions. Now, this is a hypothetical example where we're designing primers that are 4 nucleotides long, and these products are little tiny sequences. Typically, you're not designing 4 nucleotide long primers because the odds of finding any 4 given nucleotides in the genome are pretty good. So if you just design 4 nucleotide length primers, they're going to stick pretty much everywhere in the genome, or not everywhere, but lots of different places. Instead, in practice, you're typically making primers in the area of about 20 nucleotides or so, because the probability of that landing at a random spot in the genome is pretty small. The other thing is we're going to make 2 primers. They're going to be facing each other in terms of their 3' hydroxyls. And so you want their 3' hydroxyls facing each other, so when they land on their templates, they can synthesize going like this, and we're just copying this piece of data. We don't want our 3' hydroxyls pointing out that way, because then we're kind of copying away from what we want to actually amplify. Okay, so now the other thing is these products are pretty small, usually not PCRing such little tiny things, but for this application, for this example, it works well enough. So now the PCR process is, remember what we're going to do is we're going to cycle through a bunch of different temperatures. We're going to put in a test tube. We're going to put in a template DNA, so the sequence we see here at the top. We would add our primers, the things that we're going to design here. We'd add Taq polymerase. Taq polymerase is a thermal stable DNA polymerase that can stand being heated up to a high temperature, because we're going to heat the sample up to nearly boiling, about 95 or 96 degrees Celsius. Then we cool it down, so that first heating up to 95 or 96 degrees Celsius is what we call the denaturation step. That's where we're going to break all the hydrogen bonds between the double-stranded DNA, and that makes all the DNA in the test tube single-stranded. We then cool that reaction down into 55 to 65 degrees, depending on the primers, so that the primers can anneal or stick to the spots of the template DNA through complementary base pairing. Then we heat it up a little bit more to 72 degrees. That's just the temperature that Taq polymerase likes to work at. So that's our elongation step where Taq polymerase is actually going to synthesize. And we just do that over and over and over again, 25, 30 times. By the end of it, you start with your small amount of DNA to start with. You end up with billions of copies of your target sequence. Okay. So now, for this particular example, we're going to, each one of these bold regions, we're going to design primers for. So A, for segment, this spot here, primer A, primer B, primer C. We'll determine the length, and then we'll try to figure out which ones of these particular fragments are the correct one, or which one's which. Okay, and so so let's have a go at it. All right, so now I wrote down the sequences down here. Now, when you're designing primers, what you want to do is your primers are going to go to the edge of the sequence that you're trying to capture. Okay, so when we're trying to capture here, we want to get this ACT at the beginning. We want to go all the way to that A. And this AAA, we want to go all the way to that last A. So that full sequence, that's going to define the bounds of where we want our primer. Just like in B, we want that T at the one side and that A at the other. And so I just wrote these sequences down here just to kind of take them out of context and make it a little bit easier for us to see. And so the first thing you do when you're designing primers like this is the easiest thing to do is to write the second strand in, because once you write the second strand in, your primer sequences are there. You will be able to see them because they're in your sequence. Okay, so these are going to be 4 nucleotide or 4 base pair long primers. So let's first write our second strand. So it's anti-parallel. So we start with a 3. We go T G A G T T C T T T G T A C G G C T T T 5 prime. Okay. So now, when we're looking at this, what we want to remember is that we want our 3' hydroxyls facing each other. Okay, and that whatever sequence we choose, it's going to bind to the other strand by complementary base pairing. So if I pick those 4, they're going to stick by complementary base pairing there. Whereas if I pick those 4, they're going to hybridize through complementary base pairing there. And if they hybridize the complementary base pairing here, that means that is the primer that DNA polymerase is going to use, and it's going to use the top strand as the template. So if it's using the top strand as a template, this primer is going 3 to 5. So over here is where our hydroxyl is. So DNA polymerase is going to start synthesizing DNA that way. Okay, we don't want that. What we want is the 3' hydroxyls pointing in the direction of the product that we're trying to make. And so that is not the sequence that we want. We don't want that sequence, which means we want the other sequence. Okay, so here, if we chose this other sequence, ACTC, it would hybridize here. We'd have 5' and 3'. There's our hydroxyl. And so now DNA polymerase can use the bottom strand as a template and be able to synthesize DNA going in that direction. Okay. Similarly, if we go over to the right-hand side, it's either GAAA or it's CTTT. Okay. If I pick GAAA, well, that's going to stick to this bottom strand, use the bottom strand as a template, and go in the direction of the 3. Okay. So if I pick, so I don't want that. That's the other option is TTAC, where we're going 5' to the 3'. So if I pick TTAC, TT, or TTTC, we have 5' to 3'. There's our hydroxyl, and we can synthesize that. Okay.
So ACTC is one primer, TTTC is the other primer. Now, keep in mind, by convention, DNA sequences are written in the 5' to the 3' direction, and so this would be written 5' T, 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 C, 3'. Okay. Now we'll write all these up into the top part in just a second. And so now for this one, again, we would just write the other strand. We have A, T, A, C, A, T, A, C, C, T, 5'. Okay. Again, so kind of one trick you could kind of go with is we have the 3 over here and we have the 3 over here. Okay. So the 3, this top strand, the 3 is on the right. So I need to have that on the left because I want it to go right. So if I pick those 4 sequences, they would bind here, 5', 3', and they'd go like that. Okay, and since my 3' is over here, if I pick these 3 sequences, I need to go A, C, C, T, 5', 3'. And we go that way. So I have 5' T, A, T, G, 3', and I have 5' T, C, C, A, 3'. Okay. And the last one, just more of the same. Okay. And so this one, we're just going to take our, let's be consistent, 3', and we go G, T, A, C, T, 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 C, A, G, A, G, T, G, A, 5'. Okay. Again, if I pick this bottom sequence, it would land up here, and I'd be going that direction. So I don't want to do that. What I want to do is I want to pick this top sequence, so I go C, A, T, G, 5', 3', so I can synthesize that way. And then if I picked, say, that top sequence, it goes C, A, C, T, my 3' would be there, and I'd be going that direction. I don't want that. So instead, we pick the bottom sequence. In the bottom sequence, we go G, T, G, A, 5', 3', and I'm going to go in that direction. All right, so if I summarize those and put those into the table up above, we have 5', 5', A, C, T, C, 3'. Here we have 5', T, 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 C, 3'. This one we have 5', T, A, T, G. These are all the sequences we just wrote down below. 5', T, C, C, A, 3'. And this would be 5', C, A, T, G, 3'. And then the last one would be 5', A, G, T, G, 3'. Okay, so now the next question is determine length of the PCR products. Well, the length of the PCR products is the length from primer end to primer end, which is just from say, here to here, and from here to there, and from here to there. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 20 nucleotides or 20 base pairs. B would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. That's 12, and this would be 1, 2, 3, 4, 5, 6, 7, 9, 10, 11, 12, 13, 14, 15. And C would be 15 base pairs. So now let's take a look at our gel. All right, so now your lab mate must run out your primers in a PCR to see if they work. They set up PCR to visualize the products and run some reactions on the gel. I can't remember which reaction is which. Using your expected size values can help your lab mate out, and which lane represents A, B, and C. Which lane represents A, B, and C. Explain to your how you know which lane is which reaction and what the other two lanes show. Okay, so here we have four. This is our size standard or our marker or our ladder. We have 4 lanes with bands in them and then 1 lane with nothing in them. Okay, so this one with nothing, this is what we call a negative control. Whenever you do PCR reaction, you always have to include a negative control, which is all of your reaction components, your TAC, DNTPs, primers, everything except your template DNA. Okay. And the reason for this is you want to make sure that none of your reagents are contaminated with foreign DNA. And because otherwise you don't know what you're amplifying. And so if you ever have you run a tube with no template DNA, if you ever have a band show up in that tube, well, you know your reagents are contaminated. You got to throw the whole thing out and start over. Okay? And if you have any contamination in negative control, none of your results are valid. You got to start over. Okay. So the last lane is a negative control. And then what we're looking for is we're looking for a 20, a 15, and a 12. Okay, and so here we go. And so if we look right here, here's 15, there's 15. So this first one must be product C. This must be C. Okay, and then the next one, we see 20 to 20. So this next one must be product A. Now this next one is landing in between 15 and 20. We don't have any that lands in between there. So that's a question mark. All right, and then this last one is right here. It's a little bit above the 10, so that must be B, which is 12. Okay. And so what are the other 2 lanes? Well, this last lane, that's negative control. And lane 4 must be that there must be your PCR products. They maybe put the wrong combination of PCR primers in, and they're maybe amplifying another region of the genome. It's some unknown product. It's not what you're expecting. So whenever you're doing PCR in the lab, you're always knowing what your anticipated product size should be, and then you always check it on a gel to make sure that you got that product size. And if you get a product size that is not what you expect, it means that your experiment is not, you're not amplifying what you think you're amplifying, in which case you typically would try it again. You maybe sequence that product, see what it is, see if maybe you got some inserted DNA in there, but most likely you've just amplified the wrong thing. And the solution to that would be to design new primers and try to capture a different segment and try to rearrange things a little bit and capture a different piece of DNA that still catches what you're trying to catch.
So overall, this is how you would design primers, and what those primers would look like if you ran the products out on a gel. We always use gel electrophoresis as just kind of the follow-up to pretty much every step in the lab to verify that our sample's still there and that our sample's intact, that it's not degrading, that it is of the size that we expect it to be before we move on to the next step of any experimental procedure or flow of experiments. It's always, the gel's always kind of the double check that we're always going to check. So hopefully this helped, and with that, have yourself a great day.